Hi, everyone. My name is Rawan Jarrar, and I worked with Dr. Isabel Bouard from the Neuromagnetic Lab at Anschutz to study the neural correlates of selective attention and visual conflict monitoring using a Stroop task. I'd first like to thank my Capstone committee, Dr. Bouard, Dr. Thompson, and Peter Thiel, as well as Colleen Monahan, Dr. Shuddick, and Dr. Sutton for their amazing contributions to this study. And of course, I'd like to thank the Modern Human Anatomy program. So let's start by defining what a Stroop task is. So when we look at these words here, we can see that the word and the ink that they're written in match. This is a congruent stimulus. With the Stroop task, we see that the words and the color do not match. This is an incongruent stimulus. So the goal of the Stroop task is to try to identify the color and ignore the actual word itself. This has been a paradigm that's been studied for decades, and it's consistently been found that identifying the color of an incongruent stimulus versus congruent has results in a longer reaction time and an increased error rate. So the Stroop task can be used to uncover the neurological architecture that underlies executive functions. This can be from decision-making to mechanisms of control. It also has implications in psychiatric and neurological disorders, including ADHD, schizophrenia, dementia. These individuals tend to have problems with attention, and the Stroop task has long been a measure of how well these individuals can focus their attention. It also has implications in cognitive changes throughout the lifespan. It plays a major role in understanding selective attention, which is how the brain filters information and prioritizes what is important and what is not. This is related to conflict monitoring, which is how the brain recognizes and resolves conflicting pieces of information. These two processes are the focus of our study, and they're thought to occur via top-down processing. So top-down processing is when information is initially processed and interpreted in the frontal lobe, followed by signaling in posterior regions of the brain. Now, in order to confirm that these processes are occurring in a top-down manner, we need both spatial information, so where in the brain is this activity occurring, as well as temporal information. So what is the chronological order of this activity? This leads us into our methods. We use magnetoencephalography, or MEG, to measure the brain activity of participants while they did a Stroop task. Here's an example of an MEG. It's very stylish, as you can see. So the way that MEG works is that when a neuron fires, it creates an electrical current. And when a large group or cluster of neurons fire together, the sum of their electrical currents creates a magnetic field. And this is what MEG is measuring. These magnetic fields are very small, very tiny, and so we need highly sensitive magnetometers called superconducting quantum interference devices, or SQUIDs. There are about 248 SQUIDs above this man's head, and they are able to detect this outer magnetic field that we can later kind of trace down to its estimated source. The SQUIDs provide us information directly from the brain as waveforms shown here. So past studies using the Stroop task have included EEG. EEG has excellent temporal resolution on the millisecond timescale, but is not as spatially resolved as modalities such as fMRI or PET scans. These neuroimaging modalities have excellent spatial resolution, but are less optimal in their temporal resolution. So this is where MEG comes into play. It has temporal resolution in line with that millisecond timescale that EEG has, but it has the added benefit of being more spatially resolved because it's directly measuring those magnetic fields. So our study design involved 28 subjects. While they were in the MEG, there was an auditory cue of either word or color, followed by our visual stimulus. We had our incongruent stimulus where the color and word didn't match, the congruent stimulus where the color and word did match. We also had a word reading task using black ink, and we also had the color identification task using neutral words and our specific ink color. We then measured the brain activity during that reaction time and then looked at the first 1,000 milliseconds post-stimulus onset. The raw MEG data was then inputted into Brainstorm. It's an MEG analysis program that's fully interfaced with MATLAB. And we then took the data from our 28 subjects, we processed it, filtered it, removed any artifact or bad filters, and then averaged the data. The waveforms here that are detected at the sensor level can then be kind of reverse engineered or traced back to their estimated sources in the brain using minimum norm estimation, or MNE. MNE allows us to then visualize these estimated sources on a cortical model as shown here. We can then run our paired t-tests. We did two comparisons, incongruent versus congruent and incongruent versus neutral. These are also separated by task type, word reading, and color identification. For the purpose of this shorter presentation, we're going to focus on color identification since that's more relevant to the Stroop task. And then within that first 1,000 milliseconds, we divided it into 100 millisecond time averaged windows. And then finally, we were able to create statistical maps based on the maximum t values from these t-tests. So our hypothesis involves two regions of interest, the left anterior cingulate cortex, or ACC, and the left dorsolateral prefrontal cortex, or DLPFC. These two regions have popped up pretty consistently in past studies of the Stroop task. The ACC is thought to play an important role in selective attention, and the DLPFC is thought to be involved in attention switching and also inhibiting inappropriate responses. So it's also heavily theorized that the ACC conveys information to the DLPFC, followed by information posterior regions. So for our color identification tasks, for both comparisons, we hypothesized that the ACC would precede activity in the DLPFC, followed by activation in the posterior regions, in line with top-down processing. So for our first comparison of incongruent to congruent, we found significant differential activity between 700 and 1,000 milliseconds post-stimulus. We can see in this time window here, activity in the DLPFC, as well as the temporal parietal junction, or TPJ. Then took this time window and conducted additional t-tests with 10 millisecond average time windows, and saw that in this 10 millisecond window, we still see that dual activation of these two regions. We then took it one step further and got a 3 millisecond average time window and can continue to see that dual activity. And that's the beauty of MEG, that we can see such a small time window. For the incongruent neutral comparison, we found significant differential activity between 600 and 900 milliseconds post-stimulus. Initially, we see activity in the superior frontal gyrus and what appears to be some activity in the ACC, but these regions are so close to each other that it's difficult to distinguish between the two. Later on, we do see more clear activity in the ACC. We also see activity in the extrastriate. This is part of the visual cortex and is involved in visual processing. And then again, we see activity in the temporal parietal junction. The TPJ may play a role in attention. It might even be receiving information from the external environment directly and transmitting it elsewhere in the brain. So to conclude, for our first comparison, there was no differential activity in the left ACC, and the left DLPFC activity did not precede activity in posterior regions. Instead, we saw simultaneous activity in the DLPFC and the TPJ as early as 700 to 703 milliseconds. In our next comparison, there was no differential activity in the left DLPFC, but we did see what looked like activity in the superior frontal gyrus and the ACC preceding activity in the left TPJ and extrastriate. So these findings might call for a reevaluation of the functional networks involved in these processes. They may or may not be strictly in line with top-down processing. So we are definitely in need of a better framework for these processes. This is important because once we have a stronger guideline, we can use that to detect deficits in the brain from injury, from aging, or other neurological disorders. So in the future, I think there are other task switching paradigms other than Stroop that would definitely benefit from the use of MEG with that amazing temporal resolution. And then also it might be interesting to combine EEG with fMRI to get the temporal resolution of EEG and the spatial resolution of fMRI for a more robust study. That is it. Thank you so much for your time, especially everyone that stayed for the whole session. You guys are troopers. I will open up the floor to any questions.
Excellent work. Very cool presentation, Rawan.
Thank you.
So I guess I'll jump in with the first question. What was one of the alternative task switching paradigms that you might use?
Yeah, so there's a couple. There's like the flanker test. There's like a stop no go, a Simon task. These are all just kind of different variations of kind of incongruent and congruent stimuli. So they're all kind of just different variations of each other.
Great, thank you.
Yeah.
Any other questions from the gallery?
Yeah, I have a question. Rawan, why does MEG have better spatial resolution than EEG?
Oh yeah, that's a good question. So the best way that I can understand it and explain it is that so MEG and EEG are essentially locating the same source, just in a different way. So EEG is getting the electrical current, and when the signal from an electrical current can become distorted as it travels through different layers of the brain. So it has to go through the rest of the cortex, the scalp, the skull, the scalp to reach those electrodes that are placed on the scalp. With MEG, the electromagnetic field, it doesn't get distorted when it goes through different tissues, so it produces a more accurate reading. So that's why it has better spatial resolution.
Thank you.
Hey, Rawan, I have a question.
Mm-hmm.
Do you expect the top-down processing to be impaired in attention deficit disorders or any disorders where attention is being impaired?
Yeah, I would definitely, if these processes like selective attention are in fact working in a top-down manner, I would expect that we'd be able to see some sort of deficit in individuals with any sort of neurological psychiatric disorder. We may not see that like clear representation of a top-down manner, but it would be, I guess, as of now, it'd be difficult to. We don't really have the greatest like baseline, since it's still kind of up in the air of exactly how these processes are working.
Thank you.
I have a question.
Yes.
So I noticed in the in your presentation, it was the colors were red, green, and blue.
Mm-hmm.
Now, I mean, is there a version of this test that is accessible for people with deuteranopia? And if so, are the outcomes the same or different?
Are those individuals who are colorblind?
Red, green, colorblind. Red, green,
Oh,
colorblind. Yeah.
That's a really, I'm not sure. That's a really good question. I would imagine it wouldn't be terribly difficult to make a variation of the Stroop task with different colors that someone who is colorblind would be able to distinguish a little bit more easier. But I'm not sure. That's an interesting question, though.
All right, any final questions?